And I'm delighted to introduce Professor Charalambos Kyriacou, who's Professor of Behavioural Genetics at the University of Leicester, UK. So Bambos has a remarkably broad background and interests, having taught on subjects ranging from molecular biology to medical genetics through to social psychology. He's held prestigious positions in a number of universities in the UK and spent a period as a postdoc in Brandeis University in Massachusetts. He's a recipient of several prizes, most recently the Oxford Sleep and Circadian Rhythm Neuroscience Institute Prize in 2017. And he's been interested for many years in circadian rhythms and molecular analysis of clock genes using Drosophila as a model organism. And he notably worked on clock genes with Jeff Hall and Michael Rosbash, who went on to win the Nobel Prize in Medicine and Physiology for this work in 2017. He became interested in neurodegeneration around 15 years ago, working on HD alongside one of our previous SBAT members, Flavio Giorgini in Leicester. So thank you very much, Bambos, for doing us the honour of opening this year's meeting, and we're very much looking forward to your talk.
Thank you. Let me share my screen. Can you see that?
Yes, that's perfect.
Okay, so this is from biorhythms to Huntington's, and it's a story really of two boys, their mother and stepmother, a rock star, and two extraordinary coincidences. So why am I here? That's me, in case you can't see me. Patrick visited our department a couple of years ago. He was visiting my colleague, Flaviano Giorgini. Yeah, I think he was doing a thesis or he was giving a talk, I can't remember, but he came to chat to me about my work in biological rhythms. And during that conversation, I told him about my family history and how it was relevant to Huntington's. And a few months later, he invited me to the Bologna meeting, which sadly was cancelled because of COVID. So how does my geneticist working on circadian 24-hour rhythms get into Huntington's disease research? Well, let's start at the beginning and a little bit about me. I got my degree in psychology at Birmingham. There's me all those years ago. I did a PhD in genetics in Sheffield. There's me getting my degree. I was a junior lecturer in psychology in Edinburgh. And then the most important part of my education was going to become a postdoc at Brandeis University, which is a Jewish university in Boston, Massachusetts. I then returned to the psychology department as an independent fellow in Edinburgh. There I am. And then in 1984, I got my first real job as a lecturer in behavioural genetics at Leicester. And there I am with a couple of my football mates from the department. Now, that's Jeff Hall, and I did my postdoc with Jeff. Now, Jeff was a student of a remarkable man called Seymour Benzer. In my opinion, Seymour Benzer is the most brilliant biologist never to win the Nobel Prize. He was a physicist originally, and he helped develop the transistor, which his colleague Shockley won the Nobel Prize. He then moved on to genetics, and he did some pioneering work in viral genetics that laid the foundations for the triplet code and our understanding of genes and mutations. And he should have got the Nobel Prize for that, but he didn't. And then in the late 60s, early 70s, he turned his attention to behavior and the nervous system using the fruit fly as his model organism. And his idea was to use mutations as a surgical dissection instrument to dissect the nervous system and see how the nervous system controls behavior. And Jeff was one of his students. And I joined him in 1978 with my own money from the UK to do a project that was really extending my PhD. Now, Jeff was a very interesting guy to work with, a little bit on the spectrum. He never used to confront us when he was angry with us, but he used to leave us notes like, when using tools from this drawer, please replace them as soon as you're done, God damn it. Do not allow this thought ever. Leave these pens here, damn it. Stop taking pens without returning them, God damn it. So he was an interesting character to work with. And within about 6 months, I had dumped my original project because I discovered something interesting in the courtship display of fruit flies. Now, that's a male and that's a female, Drosophila melanogaster. And unlike male undergraduates, the male doesn't jump on the first female he sees. He serenades her with a courtship display. He sticks out his wing and sings her a love song, licks her bottom, and then attempts to copulate. And what I discovered was a nice rhythm in the song of the male fly. And then I wondered whether the rhythm mutations that Seymour Benzer had worked on a few years earlier might affect my courtship rhythm. Now there's Seymour Benzer again, and this is one of his PhD students, Ron Konotka. In the late 60s, Ron and Seymour decided they wanted to look at the genetic basis of circadian rhythm. Now, circadian rhythms percolate through every physiological, behavioral, and biochemical activity of every organism that lives on the surface of this planet. Everything cycles, and that's because, of course, we've evolved on a rotating planet with a period of 24 hours. So it's a fundamental. I remember at school, I was taught that a living organism respires and reproduces, the 2 R's, but also there's a third R, and that's rhythms. All animals and plants have rhythms. So what Ron did, he did a mutagenesis. He fed flies a chemical mutagen, and then he looked at their locomotor activity rhythms, or their sleep-wake cycles. This cartoon shows you one day, the second day, the third, and up to the 5th day of the locomotor behavior of a fly. And he's in a tube, and there's an infrared detector which detects when he moves. So the fly sleeps, then he wakes up, then he sleeps again. On the day 2, he wakes up at exactly the same time, 24 hours later, falls asleep, and so on and so forth. So a normal wild type fly has a period of 24 hours. Now, Ron found a mutant that had a short, fast rhythm of 19 hours. This guy wakes up 5 hours earlier on every successive day. Another mutant had the opposite phenotype. In other words, it woke up 5 hours later on every successive day, so it had a slow clock, 29 hours. And this mutant was insomniac. It was arrhythmic. And when Ron mapped the 3 mutants back, as you do in Drosophila, he just did genetic mapping, they all mapped to the same spot on the X chromosome. So they were all mutations, different mutations of the same gene. And he called that gene period for obvious reasons. So period short is a mutant with a fast 19-hour rhythm. Period long has a 29-hour slow rhythm. And period zero is arrhythmic. So this was the first demonstration of the behavioral gene, because even in zero, the arrhythmic animal, the animal is fine, it's healthy, it's just not rhythmic. And up to this time, which is 1971, nobody really believed that single genes could control behavior. It was remarkable. So Jeff knew Ron because they were both at Caltech together with Seymour, and he asked for the mutants, and Ron sent the mutants, and I found that these mutations also affected my courtship rhythm. So I'd done my original project, and I just started working on period for the next 2.5 years. Now, Jeff's best friend was Michael Rosbash. He was a molecular biologist, also at Brandeis. He'd been in Edinburgh while I was in Edinburgh, but we'd never met. But he became a good friend of mine, and we used to go out and watch sports together. Now, before I left in 1981, I had a chat with Michael. I said, Michael, I think it's time that you and Jeff got together and tried to molecularly identify the period gene, because it's not been done before. And I think it would be a good idea. And the tools are there now. And he said, yeah, great idea. And why don't you stay in America and do it? I've got lots of money. And I said, well, actually, we just had a baby. I've got a job in Edinburgh waiting for me. So I had to leave. And that, in retrospect, was a very bad decision on my part. So I left, but we kept in touch. And indeed, a couple of years later, Jeff, here he is, and Michael, they recruited some talented postdocs, and they started attempting to molecularly clone the period locus. At the same time, Michael Young, who was at Rockefeller University in New York, had the same idea, because he'd done his PhD on that region of the X chromosome that includes period. So he was looking for a gene to identify molecularly using the new technologies that were available in Drosophila. And he recruited Ted Bargello and Rob Jackson, who actually became good friends of mine, particularly Rob. And the two groups competed. Now, in America, when you have a competition, it's a competition. The two groups didn't really talk to each other. But of course, Rob and I were very good friends, and I'd actually helped Rob with his thesis. So we kind of kept each other informed. And every time one group published, the other group published at the same time. And this became very important. And the key papers were in 1984, was this paper by the Brandeis group, and it's published in December 1984. And this paper from Michael Young's group was published in Nature, also in December 1984. And this these experiments showed that if you take a piece of DNA that you think is from the period locus and you transform it into the genome of an arrhythmic per0 mutant host, if that piece of DNA carries the period sequences, then you should restore rhythms. And that's what both groups showed. So in other words, we had rescued arrhythmicity in a per0 mutant host by putting in the period sequences. And that was a kind of a big deal. In the next 20 years, saw the Brandeis and Rockefeller groups consolidate this work and build a model of the clock. And they identified by mutagenesis a lot of other clock genes, which I'll tell you about in a second. And I collaborated with Jeff and Mike for about 20 years on various projects, but mostly on the evolution of clock molecules. So I kind of embroidered the margins of the field. So how did Jeff, Michael, and Mike demonstrate how the clock works in flies? Well, here's the period and the timeless genes. And they're activated early at night by two proteins called clock and cycle, who sit on the regulatory region of period and timeless, it's called an E-box, and activate the messenger RNA early at night. That's translated into period and timeless proteins. Along comes casein kinase 1 epsilon, or double time. It's a kinase, and it sticks phosphorus groups on PER, and that identifies it as a target of degradation. So PER is degraded. Later at night, PER levels, what PER molecules are available bind to timeless, and this protects it from phosphorylation. PER and time dimers build up late at night. They move back into the nucleus of clock cells, and they block the actions of clock and cycle on their own genes. So they negatively auto-regulate their own genes. It's a negative feedback loop. When the lights come on at dawn, a photoreceptor called cryptochrome is activated, and that interferes with timeless and period, and they're degraded, and that removes the block on the transcription of period and timeless. So now clock and cycle can restore the cycling or the messenger RNA of period and timeless, and the whole cycle begins again. So it's a very simple model where you've got period and timeless as negative regulators. They regulate their own genes via clock and cycle that are the positive regulators. You have a modulator, casein kinase 1 epsilon, and a photoreceptor. And that's basically the clock in the flies. But who cares about flies? Nobody cares about flies, but you do care about mammals. And in mammals, the work of Joe Takahashi and Stephen Reppert, about 10 to 15 years later than the fly group, showed almost exactly the same molecules were involved in the mammalian clock, and that means the human clock. One evolutionary quirk is that timeless has been replaced by cryptochrome. The cryptochrome is a photoreceptor in the fly, but it's actually a negative regulator. It's taken the role of timeless in the mammal. And the circadian photoreceptor is called melanopsin, but you've got casein kinase 1 epsilon, PER and CRY are negative regulators, and clock and BMAL1, and BMAL1 is just another word for cycle, are the positive regulators. So the clock in mammals and flies is conserved. I have to say the clock in the fly is a little bit more complicated, but we don't need to go into that. Now, if you take some families have circadian and sleep disorders, and some of these disorders actually map to the period and casein kinase mutations. So these morning larks that get up early and go to bed early have mutations either in their period gene or their casein kinase gene. There are also night owls, and these people have mutations in another period gene. There are actually 3 period genes in mammals, and also in casein kinase, and also in cryptochrome as well. So the work on flies has had ramifications. It's generated a model for the clock that's conserved in mammals, and it has implications for human sleep and circadian dysfunction. And in fact, the period and casein kinase mutants all affect PER, phosphorylation, and stability, and that generates circadian and sleep disorders. And this was a big deal. How big a deal? Well, the 3 guys, Jeff, Michael, and Mike, won the Nobel Prize in 2017. And there they are about to get their prize from the King of Sweden in Stockholm. And there's some of the, I was invited to that amazing weekend we had in Stockholm. I tell you, Swedes really know how to party. And there's Mike Young, who became a very good friend of mine, and Amita Segal, another good friend. She was his most illustrious student. She identified timeless. And there's me and my wife, Susie. As you can see, we scrub up very nicely. There's Michael with Susie. There's Jeffrey, Susie. They didn't really want pictures with me. I wonder why. But we had a wonderful time. We even missed our flight home. Okay, so let's go back a bit to 1974 to 1987. I was married to Celia, who I met in Sheffield, and we were in America together. And just before we left, we had a baby, Alex. He was born in 1981, and there he is at the age of 21. And we went back to Edinburgh, and then we moved to Leicester, and Stephen was born in 2005. Now, around 2003, I had this phone call from Celia. We'd been divorced about 16 years by then, but we were still friendly. And she was very distressed. She told me that her mother, June, had been diagnosed with Huntington's. And June was to die in 2007. Now, June had two daughters, Celia and her older sister, Julia. And sadly, both of them were positive for Huntington's. So that left me with my sons, Alex and Stephen. Both had a 50% chance of inheriting the Huntington's gene from Celia. Now, sadly, Celia died about 5 years ago. She didn't die of Huntington's, she died of pancreatic cancer. She was 62 and she was just beginning to show some signs of Huntington's in terms of her motor coordination. So I had this cloud around the future of my sons, Alex and Stephen. So I got interested in looking at Huntington's research. And I knew a little bit about Huntington's gene. Being a geneticist, I knew about the number of repeats. If you have 40 or more, you tend to show the disease. I also knew that the longer the repeat, the earlier the age of onset, that there's a huge amount of variation in the age of onset. And 10 years ago, about 40% of the variability in age of onset was due to other genes, genetic modifiers. Back to Seymour Benzer. Now, there he is in the 1950s, very handsome, and he did this very interesting study in 2000. He'd taken Drosophila and he'd given them Huntington's disease by expressing in their genome 100 polyQs, these CAG repeats. And the flies develop Huntington's, and their motor coordination goes with age, and they show neurodegeneration. But what he did, he mutated genes, other genes in Drosophila, to see if he could suppress the phenotypic effects. And he found a number of genes. And this is Flaviano Giorgini. And in 2005, he published a paper where he took the same strategy, but instead of using flies, he used yeast. Basically, he gave yeast Huntington's, and that kills yeast. But by mutating other genes in yeast, he found some genes, some mutations that would rescue the viability of the yeast, as you can see here. And one of these genes was the gene for KMO. And KMO is the gene that encodes yeast kynurenine 3-monooxygenase. And that would rescue the deleterious effects of sticking in 103 polyQs into yeast. That was very interesting. So if we look at the pathway in which KMO is involved, it's the tryptophan pathway, which eventually produces serotonin. And there's KMO, there's the enzyme. And the mutation in yeast was blocking kynurenine 3-monooxygenase. So 3-HK, 3-hydroxykynurenine, and the downstream metabolites in the mutant will be reduced to nothing. And you've got this pathway to kynurenine aminotransferase, which makes kynurenic acid, which I'll call kyna. And with the mutation, you shunt kynurenine to kyna. You get more of that. And that's apparently what was saving the yeast, the Huntington's yeast. Now, there's extraordinary coincidence, number one. So Flav applied for a job at Leicester, and I interviewed him. And I was very interested in his work because of, I was interested in Huntington's research. And in fact, in my own naive way, I wanted to get into Huntington's research. Now, he was married to Nicola Suter. And amazing coincidence was that Nicola, I knew, Nicola had been a PhD student in biochemistry department in the mid-90s, upstairs from us in genetics. In fact, some of my male postdocs really fancied her. Anyway, Flav turned up with Nicola Suter. And the other thing about Flav that a lot of you don't know is Flav is an actual rock star. He had a band in the 1990s called Squirt Gun that had top 100 hits, and they had videos on MTV, and he's a lead guitarist. He writes the songs. That's his brother Math on bass. And some of his music has actually been used for movies. There's a movie called Mallrats, a cult movie, and he wrote the music for that. And it was produced by Ben Affleck. So he's a bit of a star. So Squirrel Pond were kind of famous band in the mid-90s. They toured in big stadia. The two bands that used to support them were Green Day and Blink-182. And some of you in the audience might know that those two bands became world famous sometime later. And then he started another band called the Simians. But who's the drummer? And this is the other extraordinary coincidence. The drummer was a guy called Ori Schaefer, who I'd known for 20 years. He's another fly geneticist that works on circadian rhythm. What two extraordinary coincidences. So it was fate that Flav and I should meet up. They made an album, Simians. It was marginally successful, pretty cool. And I managed to get some of their publicity shots. There's Flav and there's Ori looking cool, the cool undistracted look, the cool undistracted look from above, the cool walk with slightly different orientation of Ori and Flav, the cool undistracted from below shot, the tough guy cool shot. And then this is the constipated robot shot, which really is not cool at all. I don't know what Ori is doing there. And then there's, of course, the obligatory late night cool studio shot. So Flav was a real rock star, and I was kind of starstruck by him, but he was actually a wonderfully nice guy. And we got on like a house on fire. We have the same kind of similar pure sense of humor, I guess. And there's Flav. So Flav wanted to get a grant. He wanted to take the yeast work into mice. But in England, to get a grant, the agencies are very conservative, and he wouldn't get money for that. So I said, Flav, why don't you do the Seymour Benzer trick and then take the yeast system first into flies, which are about halfway up the evolutionary scale to mice, and then you might get a grant to work on mice. So we got some money from HiQ for 1 year to see if we could move the yeast system into flies and look at KMO mutants in flies. But Leicester is not a very glamorous place. It's a grimy Midland city. It's only famous for genetic fingerprinting from my department, Richard III, who was discovered in the car park a few years ago. And of course, Leicester City won the Premiership a few years ago. But nobody applied. So we were kind of desperate. And I knew someone who might be interested. And that was another Italian called Susanna Campuzano. Susanna was a fly geneticist, and she was also my wife. And she and I had done our own genetic experiments. There's parent one, Italian, I'm Greek, and here are Grits, Ricardo and Sophia. And she'd taken care, she'd given up her academic career. She didn't want an academic career because it's a tough job. She didn't want to be an investigator. She didn't want to apply for grants. She didn't want to give talks. She liked working at the bench. And that's difficult to do for many years. However, Flav was a good friend of Susanna's, and he talked her into taking the 1 year job. And it was going to be for 1 year only. And 13 years later, she's still working with me and Flav because the work was successful and we got a series of grants. And she's still working as a postdoc, and she likes working in the lab. And she took on the job, and it was successful. So in Drosophila, you can transform into their genome polyQ, and you get neurodegeneration, which develops with age in the fly. And in flies, for many years, we've had lots of mutants. And one of them is called cinnabar, and it changes the fly eye color from this dull red to this bright red. But cinnabar is also a null mutation for KMO. So what we did, or what Susie did, and this is her paper that was published some years ago, she looked at the tryptophan pathway in the fly. And of course, the mutation, cinnabar, would affect KMO here, and it should block 3HK and should increase kinase, just as it does in yeast. But what happens when you put the transgene with 93 polyQs? What you find is if you look at the ratio of 3HK to kinase, it increases dramatically in day 1 flies that have just been born and in older day 7 flies. So what happens with cinnabar? What we expect is that this should fall to 0. In fact, that's exactly what it does. So a cinnabar mutant, the kynurenine, the 3HK kinase ratio falls to 0, as you can see. In a heterozygote, it doesn't do anything because cinnabar is a recessive gene. So the mutation certainly does biochemically what it should do, but what about neurodegeneration? Well, To look at a fly neurodegeneration, you can look at its eyes, a simple assay. The fly has, each fly eye has 700 units. They're called ommatidium. There they are. And each ommatidium has 8 neurons, 8 photoreceptor cells. They're called rhabdomeres. And 7 can be observed under a light microscope. So if you look under a light microscope, in certain conditions, you can see those 7 cells. The 8th cell is under them, so you can't see it. And what the 93Qs do, from the Huntington gene, they reduce that number to about 4 or 5. And when you add cinnabar, you increase that number to between 5 and 6. So in fact, cinnabar does what it does in yeast. In the fly, it rescues, to some extent, neurodegeneration by this assay. Now, because Susie thought the KINA 3HK ratio was important, she also fed flies from the egg through the larval stage during their development with KINA. She just put it in the food. So the mutants will have only about 5 rhabdomeres per ommatidium. And if you feed them different concentrations, you can see you can increase that to about 5.5. You get some restoration of the neurodegeneration in the rhabdomeres. So that was very nice and was published. And of course, what we need to know is whether a KMA knockout ameliorates HD phenotypes in mice. And this paper was recently published by Gillian Bates and Flaviano. And what they did, they had a KMO knockout mouse and they crossed it into the Huntington's mouse, the R62 mouse, which, as many of you know, has exon 1 with 150 CAG repeats. So unfortunately, there was no significant improvement in motor coordination, but the periphery showed reduced levels of inflammation. And of course, inflammation is another characteristic of Huntington's, not only centrally in the nervous system, but also in the periphery. So the authors concluded, therefore, although genetic inhibition of KMO in R62 mice modulates several metabolic and inflammatory parameters, these do not translate to improvements in primary disease indicators, observations which will likely be relevant for other interventions target at peripheral inflammation in HD. So the fly work on KMO, that's the mouse work, and there is some use in the KMO work, possibly in amelioration of peripheral phenotypes in HD. So there's Susie, there's me, there's Flav, and this started a template whereby students that did a PhD with me, like Ed Green and Carlo Breda, Carlo's also Italian from Padova, just like Susie, went and did postdocs with Flaviano. And we've published a number of papers on different aspects of neurodegeneration over the last decade, as you can see here. Again, in red are the students that were trained in my lab and then went on to work in fly neurodegeneration with Flaviano. But what drove this work, what drove my interest in neurodegeneration was my sons, Alex and Stephen, and they, of course, have a 50% chance of inheriting the mutant gene. Well, there's Alex. He got into a serious relationship with a clinical psychologist, lovely girl called Holly. So 2 years ago, he had his test and he was negative. So after a COVID-related delay, exactly 1 month ago today, Alex and Holly got married. And we had a lovely day on the banks of the Thames. However, the cloud is still over Stephen. There's Stephen. That's a picture of him taken last week in Croatia, where we went on holiday. He is going to get tested towards the end of the year. Hopefully he'll be negative, but if the coin flips the wrong way, we will have to deal with it as a family, just like many people that have dealt with that are listening now. So just to acknowledge now the people that have funded the work of Flaviano and myself, which is CHDI, HDA, Parkinson's UK, BBSRC, and MRC. Thank you very much.
Thank you so much. That was absolutely wonderful. Much appreciated. Thank you for sharing that with us. I don't know if we have any questions. I think we are quite short of time. So at this point, I think we will move on to the plenary session. And I hope you'll be prepared, Bambos, to answer questions throughout the day that might come to you through the chat.
Absolutely.
Thank you again. So